Let's study 9th standard ICSE Biology chapter 9, the economic importance of bacteria and fungi. Bacteria come in various shapes and sizes. So let's learn to distinguish between them, not to discriminate between them. Cocci means spherical bacteria, bacilli, rod-shaped, spirilla, spiral, and vibrio are comma-shaped. You see all their structures. There is unity in diversity out here. If they live singly, then they don't have any special name. But if they are in pairs, it's diplococci, long chains are streptococci, and in clusters they are staphylococci. Great, now you've taken your first step towards being a doctor. Let's see how a bacterial cell looks. It may have flagellum for locomotion. It can swim with the help of these. It has a capsule along with the cell wall and cell membrane. That's a triple protection. Very difficult to kill them then, even by boiling. It has cytoplasm, of course. It has a single bacterial chromosome just floating around in the cytoplasm. Together it's called protoplasm, of course. It does not have a nucleus. It's a prokaryote. It has some glycogen granules, that is its food. Its reproduction is so simple. First of all, the DNA duplicates. Then the cytoplasm set separates. The cell wall separates them completely. And you've got two daughter bacteria. This is called fission. But how are they useful? First of all, they are helpful in making antibiotics. An antibiotic is a chemical substance produced by a living organism, usually a fungus or a bacteria, which can kill or stop the growth of some disease-producing bacteria and fungi. The discovery of the first antibiotic is quite fascinating. Alexander Fleming accidentally had dropped some fungi on a bacteria culture, and he saw that this fungus called Penicillium notatum was secreting something which could kill the bacteria. So he removed that chemical and realized that it can be used for medicinal purposes. Hence the first antibiotic penicillin was produced. Remember, Penicillium notatum is the name of the fungus. The next antibiotic was streptomycin, which was discovered by Salmon Waxman, but this time it was made from a bacterium, Streptomyces graecosus. Today, some well-known antibiotics are these, and we need to know their source organisms. So nowadays, we don't get it from Penicillium notatum, we get it from Penicillium chrysogenum. It is used against tetanus and diphtheria. We can even manufacture this chemically in the lab or in the factories. What is the criteria for a good antibiotic? It should be a broad spectrum antibiotic. It should be able to kill a variety of diseases. More the diseases it can cure, the better it is. It should not have undesirable side effects, and it should not kill the normal bacteria of the host, because there are some good bacteria, beneficial bacteria in our gut which help in our nourishment. They should not be killed by these antibiotics. There are some other uses of antibiotics as well. They are used as food preservatives, especially for fresh meat and fish. And it is even added to animal feed so that the animals, when they eat their food, they don't get diseased. It can also be used to control plant pathogens or plant germs. Next use of bacteria is serum. What's that? It's actually the blood plasma from which fibrinogen has been removed. You see, blood has the fluid part called plasma and the cellular parts, that is WBCs, RBCs and platelets. So I'm just talking about blood plasma here, not the RBCs, WBCs. They are not there. The platelets are not there. But even this blood plasma has, the liquid part of blood has many dissolved substances like fibrinogen, which helps in blood clotting. Remove that also from blood plasma. So now what is left? Serum is left. It still has some dissolved substances, especially it has antibodies or antitoxins. You see, if you get flu right now, there's a viral infection, then a war will start in your body. Your immunity will fight the virus, which will take a few days. The WBCs, especially the lymphocytes, will produce some antibodies. There are some chemicals which will destroy the germs, and you will win the war in the end. And these antibodies will remain in your blood for some time, so that you will remain immune to cold for a few months, maybe 6 months. You will not get cold again because of these antibodies. They give you immunity. That's how even vaccines work, but we'll study that in detail some other time. Now these serums which contain antibodies are useful. Imagine that your serum is used to treat somebody else who is having flu and whose own immunity is very weak. He's not able to produce enough antibodies, so he can borrow your antibodies if the blood is compatible, of course. So that's how serums are useful. But how is it related to bacteria? Well, we have been able to prepare some very useful serums by genetically modified bacteria. Yes, we can hack into the DNA of the bacteria, insert some genes so that they would obey our commands and produce whatever chemicals we want them to. For example, some genetically modified bacteria can produce blood clotting factor number 8, which can be used for treatment of hemophilia disease or even in hemophilia B disease. Another example of genetic engineering is in certain bacteria like Escherichia coli, we have changed their DNA. We have introduced a gene from the human DNA which has the code for producing a hormone, a protein called insulin. You see, we can produce insulin in our pancreas, which is very important to keep our blood sugar level low. But some people are unable to produce insulin. So they suffer from diabetes mellitus, high blood sugar level, which is very dangerous. So they need to take injections of insulin regularly. But from where will they get the insulin? Well, we have genetically engineered bacteria to start producing insulin for us. We just give it a raw materials and test tube and it makes insulin and we can bottle it and sell it. Now coming back to serum, as I said, it can have antitoxins also. Antibodies kill germs, but what about antitoxins? Well, they neutralize the toxins. Toxins are actually poisonous proteins released by bacteria or any plant or animal. So we can neutralize it with the help of antitoxins. For example, if a small dose of a bacterial toxin like that of diphtheria or tetanus is injected into a healthy animal like a horse or a cow, small amount so that it does not kill the horse or cow, but we are now encouraging the horse or cow to produce antibodies against this bacterial toxin. Next, we will remove the blood of this animal. We will remove its serum, which will have the antitoxins, ready-made antitoxins, and now we can use it for the treatment of any person suffering from diphtheria or tetanus. No need to wait for you to develop your immunity. Ready-made antibodies and antitoxins are available. Similarly, even anti-venom can be produced. Some of the snake poisons are introduced in small quantities in animals. Let them produce the antitoxins. We will use the ready-made antitoxins. That's safer for us. Although it may be cruel on the animals. But hey, what is more important for you? Saving a human life or that of an animal? Now let's talk about vaccines. A vaccine is a preparation consisting of weakened or dead germs or germ substances like toxins for developing a resistance to a particular disease. So what does a vaccine contain? It contains weak germs or dead germs or germ substance like toxins. Please understand the vaccine is very different from a serum. Serum having antibodies are used for treatment. Vaccine is used even before we are infected. It is a precautionary measure, a prevention of disease. And introducing this vaccine into the body to develop a resistance is called vaccination, also called prophylaxis or immunization. For example, if you take a vaccine of smallpox, or rather a vaccine of polio, because smallpox is not existing anymore on earth. So what is there in the polio vaccine? Well, we have the dead polio virus. Or let's take an example of bacteria. There is a vaccine called TAB vaccine, TAB vaccine, which is used against typhoid. So in that TAB vaccine, there is killed bacteria that is given to you, either orally or injected. I'm not sure about that. But once it enters your body, see, the bacteria are killed, so it won't harm you. You won't suffer from typhoid. But at least your immune system will get alerted about this kind of bacteria. If you are not vaccinated and the typhoid bacteria enter your body, then you will suffer from typhoid, which is a very dangerous disease. Because your immune system was never aware of such a bacteria. It's for the first time they're fighting a war against it. It will take time for them to defeat the bacteria. And in the meanwhile, you may even lose your life. But if you're given a vaccine, you're already giving a training to your WBCs to fight against it. You're already developing some antibodies against it. So that when some living typhoid bacteria enters your body, you are already prepared. You will win the war even before it started. BCG vaccine, BCG full form is Bacillus of Calmette and Guerin, used against tuberculosis, has living weakened bacteria. So the bacteria in the vaccine are alive, but they're weak, so they won't harm you. They'll just give practice to your WBCs to give you immunity, and then you won't suffer from tuberculosis. So that's how vaccines work. There is another kind of vaccine, the third type, called toxoids, which are nothing but the germ substances. We know what are toxins. They're the poisonous substances. Now, if these toxins are inactivated with the help of formic acid or something, and then if you introduce these toxoids into your body, so again you're giving training to your body, they will recognize the toxoids, they will produce antitoxins. For example, for diphtheria and tetanus, the vaccines actually contain not the bacteria, but the bacteria's poison, the bacteria's toxin, but inactivated form, so it's toxoid is introduced in your body. So once you produce the antitoxins, so that if ever the diphtheria or tetanus bacteria enter your body, you will not suffer from the disease because you already have the antitoxins, that is, you are immune to it. But it lasts for a short time, say around 6 months. That is why when you are injured, you may get a tetanus shot, which is nothing but a vaccine against tetanus. So this vaccine is given even before you develop symptoms, but understand that serums are used to cure the diseases. It's not a vaccine. So now, antitoxin, what's the definition? It's an antibody. It's a special type of antibody, but antibodies can kill living things also, like germs, but antitoxins only neutralizes the toxin produced by the germs. Next, bacteria's role in agriculture. They are very important in agriculture. We have some nitrogen fixing bacteria. You see, there is a lot of nitrogen in air, but the plants cannot use gaseous nitrogen to make proteins. They have to absorb it via their roots through the soil in the form of nitrates or nitrogen salts. Then they can use it to make proteins, which is very important. So who will convert this atmospheric nitrogen into nitrates and salts of nitrogen? Well, this process is called nitrogen fixation, and bacteria can help in this process, especially bacteria like rhizobium, which live in the small nodules on the roots of leguminous plants. That is why leguminous plants actually make the soil more fertile, because the nitrogen content of the soil increases thanks to rhizobium. There are some free living bacteria also which don't need any plant to survive. They live in the soil, like Azotobacter and Clostridium. Even they do nitrogen fixation. There is another category of bacteria which are also useful, called nitrifying bacteria. You see, whenever there are some dead remains of plants and animals, like their wastes or their dead bodies, etc., some bacteria will convert the nitrogenous wastes like urea, etc., into ammonia, which is then converted into ammonium compounds, which is converted into nitrites and then into nitrates. So even that increases the nitrate quantity of the soil. Now this process is called nitrification. So here we are not converting the atmospheric nitrogen into nitrates, we are just converting the dead and decaying matter into nitrates. So that's where nitrifying bacteria are also important, like Nitrosomonas and Nitrobacter. Now we have a third category of bacteria called denitrifying bacteria, which do the opposite of nitrification. That is, they break down the nitrates into nitrogen gas and release it into the atmosphere to complete the cycle. It's important that the cycle is completed so that it is recycled and it spreads across large areas. So example of this is Pseudomonas. This nitrogen cycle has to be practiced, the diagram has to be practiced. Just cancel this particular arrow, doesn't make sense. So when plants and animals die, the decay of living matter gives you ammonia, which becomes nitrites and nitrates in the soil. And these nitrates are useful for plants. The nitrates may be denitrified and converted into nitrogen, but this nitrogen is again converted into nitrates with the help of nitrogen fixation. Next, bacteria are important even in industries. For example, for developing different flavors of tea, which is called tea curing, and for leather tanning. Spoilage of food by bacteria, well, we should always avoid expired food in tins, because if the food is very old, the bacteria may have multiplied so much and they would have released some poisonous substances that eating such food will now cause food poisoning, and severe form of food poisoning is botulism. That is why preservation of food is important. But how can we preserve food for a long time? First of all, sterilization, which can be done by heating at high temperature or boiling, which will kill a lot of germs, but to kill the spores, you will need some pressure cooker heating. Salting is a common practice, especially fish are salted so that by the time they are transported, they don't decompose. You see, when the concentration of salt is very high or even of sugar is very high, bacteria cannot grow in such an environment because they will get plasmolized. That's something we'll study in 10th standard. Next, dehydration. You often keep things on your terrace to be dried before storing it in your kitchen. So similarly, grains, meat, fish, vegetables are dried because less the water content, less the chances of germs growing in it. Irradiation, we can use UV rays to kill the germs and sterilize the air in the schools, etc. Pasteurization is a technique in which milk is heated to about 60 degrees Celsius for 30 minutes and then it's chilled quickly. This will kill most of the germs but not the curdling germs. So you can still make yogurt out of such milk. The good bacteria at least are not killed by pasteurization. So it's a good alternative to boiling milk. The souring bacteria may still be present. Next, refrigeration, cold temperature. Often we keep food, fruits, vegetables inside a refrigerator because the bacteria cannot grow fast in cold weather. And then some strong concentration in chemicals. For example, mangoes rot fast, but if we convert mangoes into pickles, then we can use it the whole year because pickles have a very high concentration of salt or sugar in jams, and bacteria cannot grow in such an environment because they will undergo plasmolysis, that is, they will shrink and wither away. We can even use preservatives like sodium benzoate in tinned foods. Bacteria can cause various diseases. In plants, it can cause black rot and bacterial blight, and in animals, it can cause anthrax, tuberculosis, and in humans, it can cause so many diseases. It can also be used as a bioweapon. For example, anthrax bacteria can be used by the enemy to start an epidemic in the enemy country, which is very unethical. Now let's talk about fungi. Molds are an example of fungi. They have spores, they reproduce by spores. When the spores fall on some nutrient medium like bread, it starts growing once again. Yeast is a unicellular fungus. It has glycogen granule, nucleus, and a vacuole. It's not double tick, so you don't have to draw this. The most important use of fungus is that it helps to make wine, an alcoholic beverage. That happens by a process called fermentation. See, in our body, respiration, the glucose is broken down into carbon dioxide and water, and ATP energy is produced. But we need oxygen for that. Fermentation is anaerobic. It does not need oxygen. It can just break into ethanol, which is ethyl alcohol, and give carbon dioxide, and some energy is released. Only 2 ATP. That's enough for yeast. So that's how from fruits like grapes, etc., alcohol is produced or beer is produced. But then the concentration of the alcohol is around 12% because high concentration of alcohol is poisonous and it can kill yeast cells. Depending on the percentage of water and other substances in it, alcohol has many varieties. What are the characteristics of wine? Well, it is quickly absorbed into the body. The moment you drink it, within a few minutes, its effect is visible. It is used to improve the mood and to warm the body quickly. That's one use. But it's mostly harmful because if it is consumed in large quantities, it can cause accidents. And alcoholics may develop body ailments like liver cirrhosis, that is a first step towards liver cancer. Yeast are also used in baking to make the dough for bread or cake. Some yeast is added so that the dough rises, which is called leavening. Because through fermentation, they are releasing carbon dioxide gas, which will make the bread or the cake porous and spongy. It is also useful in making cheese, which has 4 steps. First, curdle the milk with the help of some bacteria, of course. Then remove all the moisture to make cottage cheese. Salt it so that undesirable germs don't grow, and then ripen it. The fungi added in the first step will give particular flavor to the cheese in the end. Mushroom is also a fungus and it's edible, some of them. In India, Agaricus bisporus is very popular, white button mushroom. It looks like this. Yummy, isn't it? Well, it is cultivated in 5 steps: composting, spawning, casing, cropping, harvesting, and preservation. Cropping and harvesting was step 4. But the details of this step is not in syllabus.
Hi students, this is AJ sir. If you like this video, press the like button. If you would like to enroll for my online test series or online lectures, email me or message me on Instagram. Check the description for more information.